Good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, in our previous session, we are completely dealt with the early discoveries in photosynthesis, where we have learnt all the early discoveries in a single frame. Yes, and then we have looked into the light reaction, which are also called as light independent reactions or photochemical phase. What are the three sequential reactions, beta? Three sequential reactions of light dependent reactions. First is absorption of light in the form of photons. Okay, this happens in thylakoid lamellae or thylakoid membrane, right ma? Second is what? Photolysis of water, which happens in thylakoid lumen. And third is what, Nana? Formation of assimilatory power, that is ATP and NADPH. This takes place in the stroma. Now, at the end of photochemical phase, okay, at the end of photochemical phase, what is photochemical phase? You can parallelly take down. At the end of photochemical phase, what are the products you are having, ma? Products ATP, NADPH and oxygen, right ma? So, this oxygen is evolved as byproduct, right? Oxygen which is evolved as byproduct during photolysis of water. This can easily diffuse in the chloroplast. This can easily diffuse in the chloroplast. Whereas this ATP and NADPH, this is utilized for synthesis of glucose or synthesis of carbohydrates. Okay? Both ATP and NADPH together will be calling it as what, ma? Assimilatory power, and this will be utilized for the synthesis of glucose or carbohydrates. Clear, right? Clear? Did you recall everything what we have learnt? And in our previous class, we have also dealt with cyclic photophosphorylation, non-cyclic photophosphorylation and chemiosmotic hypothesis, right? Clear with everything? Now, let us begin with the biosynthetic phase or light independent reactions. Biosynthetic phase or light independent reactions. So, calling this light independent reactions as dark reactions is a misnomer. Why? Can anyone of you explain? Calling light independent reactions as dark reactions is a misnomer. Why?
Because it takes place in night, in darkness.
It will not take place only in night. Okay.
It occurs parallelly in daytime when sunlight is available. So it is wrong that we can say dark.
Yes.
Because it tells only in absence of
light. Dark reactions exactly mean that the reactions will take place during night or during dark, but it doesn't mean the same, okay? Because these light independent reactions or biosynthetic phase are not directly dependent on light. They are dependent on the products of light reaction, and they take place both during daytime and during night time. Clear, right? Because this statement is important, ma. Calling light independent reactions as dark reaction is a misnomer. In reason, they will provide you one statement. You should be able to identify whether the reason is correct or not. Fine. Learning each and every concept in depth, in detailed way will help you to answer the assertion reason based questions and statement based questions. Okay. Now, if you have to see the biosynthetic phase, the gist of biosynthetic phase is inside stroma what happens, ma? Carbon dioxide whichever is entering into the cell through stromatal opening, this gaseous carbon dioxide will be fixed to glucose C6H12O6 or carbohydrates. Okay. This fixation requires what? ATP and NADPH, that is assimilatory power. This fixation can happen in different cycles in different plants. Okay. It can be in C3 cycle, C4 cycle or CAM cycle. Okay. C3 cycle occurs in all plants. Okay, C3 cycle occurs in all the plants. C4 cycle occurs in tropical plants. C4 cycle occurs in tropical plants. CAM cycle occurs in desert plants. Desert plants. Yes, xerophytes also you can say. So first you look into the history of C3 cycle. You can note down, I will give you time, but listen first ma, listen first. C3 cycle. C3 cycle, which is also called as Kelvin cycle, this is named after the scientist Melvin Kelvin. Okay, named after the scientist Melvin Kelvin. Melvin Kelvin has conducted the photosynthetic experiments on the algae, that is chlorella. So this is called as what? Algal photosynthesis. Okay, algal photosynthesis is oxygenic or anoxygenic? Oxygenic photosynthesis only. So after sequential experiments on chlorella, which is algae, conducting algal photosynthesis by using radioactive isotope, that is C14. C14 is what? Radioactive isotope. Using that he has conducted algal photosynthesis, and he stated that the first stable organic intermediate which is formed after fixing carbon dioxide is a 3-carbon compound, that is 3-phosphoglyceric acid. Okay. What is the result of his experiment? He said that the first stable organic intermediate formed after fixing carbon dioxide is what ma? 3-carbon compound, that is 3-phosphoglyceric acid. And he stated that C3 cycle will occur in all the plants in mesophyll cells. Clear with C3 cycle? Any doubt anywhere ma? No, right? Now C4 cycle, which is also called as Hutchinson-Slack pathway, which is named after those scientists. Okay, this is also called as Hutchinson-Slack pathway, and here the first stable product which is formed is oxaloacetic acid, which is a 4-carbon compound. As here the first stable intermediate is 3-carbon compound, this is called as C3 cycle. Here the first stable compound is 4-carbon compound, so this is C4 cycle. This oxaloacetic acid can be changed to what? Malic acid or aspartic acid in the continuation of the C4 cycle. Okay. This occurs only in C4 plants. C4 plants are mostly tropical plants. They occur in tropical climates. Most importantly, you have to remember the examples of tropical climates. Examples include most of the monocots like sorghum, maize and sugarcane. The only dicot which conducts C4 cycle is amaranthus. Okay, amaranthus is the only dicot which will conduct which cycle? C4 cycle. Clear with the C4 cycle beta? Next is CAM cycle. CAM cycle, which is also called as crassulacean acid metabolism. Here also the first stable product which is formed is oxaloacetic acid. This occurs in all crassulacean family members, okay, which grow in desert climates. As you said, the xerophytes, okay, and crassulacean family members also, that is why it is called as crassulacean acid metabolism cycle. Okay. Succulent plants like agave, cactus, opuntia, aloe vera are the best examples, and tropical epiphytes like orchids. So this is the complete description of C3 cycle, C4 cycle and CAM cycle. Is this comparative analysis clear to you? Just look at it for 1 minute, and any doubt anywhere you just ask me. What is the first stable product is important, okay, which scientist, in which plants it is occurring, what are the examples, that is it, okay, yes. So next if we have to see, first let us begin with C3 cycle. C3 cycle also called as what ma? Kelvin cycle. Named after which scientist? Melvin Kelvin. So this C3 cycle is taking place in 3 steps. First is carboxylation, second will be reduction, and third is regeneration. Okay. If there is a statement which comes in your exam, which is the most crucial step in C3 cycle? Most crucial step in C3 cycle amongst these 3 is carboxylation. Remember that. So the first step is carboxylation, where carbon dioxide enters the cell. This carbon dioxide is first accepted. What is the primary acceptor in C3 cycle is RUBP. Remember, most important. Okay. Primary acceptor of carbon dioxide is what? RUBP, ribulose bisphosphate. That is the primary acceptor of carbon dioxide. This is catalyzed by the enzyme ribosco-ribulose bisphosphate carboxylase oxygenase. This ribosco is world's abundant enzyme. Okay, ribosco is what ma? World's, see here I wrote, most abundant enzyme on earth or world's abundant enzyme. Ribosco will have equal affinity to bind with carbon dioxide and oxygen. Okay, listen here. Okay, it has equal affinity to bind with carbon dioxide and oxygen. As such, the first stable product which is formed is 3-carbon compound. So here if you see ma, carbon dioxide is 1 carbon, okay, and RUBP is 5 carbon. So on total how many carbons should be formed? 6 carbon. So 2 molecules of 3-carbon compound is formed in the very first step of carboxylation. Clear, right? Once again I will tell you, see, when Melvin Kelvin said that the first organic intermediate which is formed in C3 cycle is 3-carbon compound, then most of the scientists thought that as carbon dioxide is 1 carbon, might be the acceptor is 2-carbon compound. Then only it is forming 3-carbon compound. That was an assumption for many years, but then it is proven wrong when it is identified that RUBP is the primary acceptor of carbon dioxide in C3 cycle. Okay, C3 cycle is the only cycle which is operated in temperate plants. Of course, this is seen in all the other plants along with other cycles like C4 and CAM, but only C3 cycle, without any other cycles, only C3 cycle is operated in temperate plants. What are temperate plants, beta? Those which grow in cooler climates. Okay. So here the first step, what is that? Carboxylation, which is the most crucial step, fine, wherein carbon dioxide it is accepted by the primary acceptor. What is it? RUBP. Remember the difference between the primary acceptor and the enzyme which is catalyzing. Okay. If the question is asked about primary acceptor, you should not choose ribosco. Fine. The primary acceptor is RUBP, ribulose bisphosphate, which is 5-carbon compound. This is catalyzed by ribosco, okay, which is world's abundant enzyme. This has equal affinity to bind with carbon dioxide and oxygen. And then first stable intermediate which is formed is 3-carbon compound, that is 3-phosphoglyceric acid. How many molecules? 2 molecules. Till here it is clear, right? And the next step, that is reduction. 2 molecules of 3-carbon compound is reduced to 2 molecules of glyceraldehyde 3-phosphate. Okay? It is reduced to 2 molecules of G3P by consuming how much energy? 2 ATP and 2 NADPH. Okay? So, for 1 carbon dioxide which is fixed, for the process of reduction, how much energy is used, beta? 2 ATP and 2 NADPH. From this 2 molecules of G3P, okay, which is 6 carbon in total, right? Okay, of this 1 carbon will always escape the cycle for the formation of glucose. Remember that. Okay, from the 6 carbon, when 1 carbon is escaping the cycle, rest how many carbons will be there? 5 carbons. That 5 carbons will be regenerated back to RUBP. Okay, for this regeneration, how much ATP is consumed? 1 ATP. Okay, if there is a question, which is the most crucial step for Kelvin cycle to run the cycle uninterruptedly, if that is the question, then you have to answer regeneration. Patience, hold on, listen complete question and then answer. Sit straight. Yeah. So, clear, right? What are the 3 steps, ma? Carboxylation, reduction, regeneration. Carboxylation, what is the primary acceptor of carbon dioxide? RUBP. Okay. What is the first stable product formed? 3-PGA, that is 3-phosphoglyceric acid. How much energy is utilized? In all the photosynthetic reactions, energy will be utilized. Okay. How much energy is utilized in reduction for 1 carbon dioxide? 2 ATP and 2 NADPH. How many G3P are formed? 2 G3P will be formed. How many carbons on total? 6 carbons, of which 1 carbon will be escaping the cycle for the formation of glucose. Okay. Rest how many are there? 5 carbons, that is regenerated back, wherein this step will be utilizing 1 ATP. Clear, right? You have understood one carbon dioxide fixation, and here you have to highlight two statements. One is the most crucial step in Kelvin cycle is carboxylation. Most crucial step to run the cycle uninterruptedly is regeneration. Remember. Now, so to conduct the cycle, one carbon dioxide, on total how much energy is utilized, ma? On total,
280p.
here only I'll write. On total how much energy is utilized?
1280p.
For one carbon dioxide, I am saying, for one carbon dioxide fixation in Kelvin cycle, how much ATP and NADPH is used? 2 plus 1, 3 ATP and 2 NADPH. 3 ATP and 2 NADPH. Okay. From each cycle, one carbon is leaving for the formation of glucose. Okay. To form glucose, how many carbons are required? 6 carbons. Means how many cycles should happen? 6 cycles. How many carbon dioxides required? 6 carbon dioxides are required. How much ATP is utilized? 6 into 3. 18 ATP, 12 NADPH. NADH. Okay. Fine. You have understood completely, no, C3 cycle? Yeah. So, once you see, this is for 6 cycles. Okay, 6 carbon for the formation of one glucose. For formation of one glucose. Okay, 6 carbon dioxides are fixed. How many PGA are formed? 12 PGA. How much ATP is utilized here? 12 ATP and 12 NADPH. How many G3P are formed? 12 G3P are formed, of which 6 are leaving the cycle for the formation of glucose. Rest 10 G3P will be there. Okay, that will be regenerated to RUBP. Clear, right? And here how much ATP is utilized again? 6 ATP. Okay. Any doubt anywhere, ma? No. So, look at here. The dual nature of Rubisco you have to know. Okay. Initial first step, that is carboxylation, which is catalyzed by Rubisco, which is world's abundant enzyme. This has equal affinity for carbon dioxide and oxygen. Remember. Okay. So, if you see, if in the atmosphere there is equal, sit straight, there is equal ratio of carbon dioxide and water, okay, sorry, equal ratio of carbon dioxide and oxygen, then Rubisco will bind to carbon dioxide only. But if at all, listen carefully, carbon dioxide is to oxygen, if the ratio is nearly equal, then Rubisco will bind with what? Carbon dioxide only. But then if the ratio of carbon dioxide and oxygen is less, which means carbon dioxide is less and oxygen is more, okay, in that case Rubisco will bind with what? Oxygen. That is the dual nature of Rubisco. Fine. So, here carbon dioxide will be, it will be binding with carbon dioxide when there is low temperature and high carbon dioxide oxygen ratio. Okay. Remember one thing, the C3 cycle is the only cycle which is operated in temperate plants, I told you. Temperate plants are those plants which grow in around 25 degrees Celsius, 30 degrees Celsius maximum. So, at low temperatures, carbon dioxide will not solubilize easily. As the temperature rises, carbon dioxide gets solubilized. Okay. So, at low temperatures, there will be high concentration of what? Carbon dioxide is to oxygen ratio. In that cases, Rubisco will bind with what? Carbon dioxide. But when scenario differs, at high temperature, there will be low ratio of carbon dioxide and oxygen. Okay, there will be low ratio because carbon dioxide will be solubilized easily. Okay, in that case, Rubisco will bind with what? Oxygen.
So, in polar plants also C3 will only take.
Yes. Okay, because there will be high CO2 to oxygen ratio at low temperatures. Fix this in your mind. Okay. Now, so here Kelvin cycle at high relative concentration of carbon dioxide and low temperature, Rubisco will behave as what? Carboxylase. At high temperatures where there is low ratio of carbon dioxide oxygen, what happens is beta, photorespiration or C2 cycle is operated. Okay, photorespiration or C2 cycle. When when Rubisco is binding with oxygen, what happens? C2 cycle will be operated in temperate plants. Okay. So now let us discuss about photorespiration. Fine. What happens is, first you remember, here oxygen is being binding with what? RUBP, not carbon dioxide. Okay. And here Rubisco is catalyzing this reaction. Rubisco is binding with what? Oxygen. How many carbons are there? 5 carbons. So what it will form? 5 carbons only it will form, right? So one molecule of 2 carbon compound, that is phosphoglycolate. Phosphoglycolate, and one molecule of 3 carbon compound, that is phosphoglyceric acid. Okay. So this two will be formed, but the second step, that is reduction, will not be operated if there is one PGA. 2 PGA are must to run the cycle. So to generate one PGA more, it has to travel across 3 organelles, it has to utilize so much of energy, oxygen is being utilized, carbon dioxide will be released, and that is why, as this huge lot of process is happening, this is said to be a wasteful process. Okay. Now you have understood the concept, right? So only one PGA will be generated after fixing what? Oxygen. When RUBP binding to oxygen, one molecule of 2 carbon compound, that is phosphoglycolate, and one molecule of 3 carbon, that is PGA, is formed. To generate one more PGA, it has to travel across how many organelles? 3 organelles. Okay, and ATP is utilized. Oxygen is utilized. Okay, and carbon dioxide is released. Fine. As this whole process is happening, this is said to be what? Wasteful process, which takes place only in C3 plants. Only in C3 plants, that is temperate plants. Understood the gist of C2 cycle, beta? Now if you look into this carefully, what you have to remember, I will tell you. Okay, what are the 3 organelles? Of course, that the first step which is happening will take place in the chloroplast stroma. Okay, first organelle is what? Chloroplast. Now this phosphoglycolate which is there, one molecule of phosphoglyceric acid and one molecule of phosphoglycolate is forming, right? So this phosphoglycolate will first convert to glycolate. Dephosphorylation happens. Okay. So glycolate formation takes place in which organelle? Chloroplast. The second organelle is peroxisome. Peroxisome are small microbodies which are present inside the plant cell. We have learnt in Cell the Unit of Life, right? So this peroxisomes, which are rich in hydrolytic enzymes again. So glycolate will enter into the peroxisome utilizing oxygen. I said no, oxygen will be utilized. Okay, utilizing oxygen, this forms what? Glyoxylate. Okay, what is formed, beta? Glyoxylate is formed. Then it is converted to glycine. Glycine is an amino acid. Okay, this glycine will enter into the mitochondria. In mitochondria, carbon dioxide is evolved out. Okay, carbon dioxide is released. And here you can see NAD plus is utilized and here it is released. So it is neutralized, we are not counting NAD plus. Fine. Glycine is turned to what? Serine. Serine will enter again into the peroxisome, converts to hydroxypyruvate and glycerin. Okay. Glycerate enters into chloroplast. Here ATP is utilized again. Okay. And this is converting to what? One molecule of phosphoglyceric acid. See how much journey it is doing to synthesize one more phosphoglyceric acid by consuming so much of energy and by releasing out carbon dioxide. Yes, Kunal.
Will it get extra carbon?
Where is it getting the extra carbon?
That's what is made of.
Here it is being converted, no? Hydroxypyruvate. From here itself you can see 3 carbon. 3 carbon in serine. Okay. So from here itself the extra carbon is added. Till here it is 2 carbon beta. From here it is 3 carbon. Understood? Good doubt. Any other doubt, ma? No, right? So one thing you remember with respect to your exam, okay, from photorespiration or C2 cycle. This cycle is operated in which plants? Which C3 plants or temperate plants. Okay, this happens only when the temperature rises and when the ratio between carbon dioxide and oxygen is decreasing, low. When there is low ratio between carbon dioxide and oxygen and high temperature, live sir, live. 40 minutes more. Okay. So what you have to remember over here, photorespiration only activates in C3 plants, okay, only C3 plants or temperate plants when there is rise in temperature and there is low ratio between carbon dioxide and oxygen ratio is low, okay. So in that case what happens? Oxygen binding with RUBP in the presence of Rubisco, Rubisco will act here as, behave as what? Oxygenase or carboxylase? Oxygenase. It will form what? One molecule of 2 carbon compound. What is that? Phosphoglycolate, and one molecule of 3 carbon, that is phosphoglyceric acid. This phosphoglycolate will first convert to what? Glycolate. This is dephosphorylation. Glycolate enters into peroxisome, converts to
Dioxylate.
then to glycine enters into this, converts to serine. Serine gets enters into peroxisome again, hydroxypyruvate, glycerate, this enters into chloroplast. Phosphorylation happens, phosphoglyceric acid forms. Fine.
ATP is formed.
ATP is utilized.
Here, this side.
Where?
Phosphoglycerate.
No, it is not formed. ATP will not form over here. It is utilized. As energy is being utilized and carbon dioxide is released, that is why we say it as wasteful process.
But it is dephosphorylation rate. Phosphate is removed. This one.
When it is dephosphorylation, look, listen carefully. This is phosphoglycolate, right beta? This is the substrate. From this substrate, phosphate group is removed. And here it is simple glycolate. Here where is the concept of ATP? Here you see, ATP is being utilized. From this ATP, phosphate group is added and phosphoglyceric acid is formed. Clear, right? Okay. So this is all about photorespiration. Any doubt anywhere, beta? No. You are clear with C3 cycle, you are clear with photorespiration. Now let us look into C4 cycle. Before going into C4 cycle, you should have a clear idea on the C4 plants and their anatomy. Okay. So listen, C4 cycle occurs in C4 plants or tropical plants. Tropical plants are those plants which grow in higher temperatures around 30 degrees Celsius to 35 degrees Celsius or 40 degrees Celsius. So in this range, their adaptation, they have a one adaptation. That adaptation is called as what? Kranz anatomy. What is that, ma? First you have to understand this Kranz anatomy, then it will be easy for you to understand the mechanism of C4 cycle. First you look at here, vascular bundles whichever are there, you see here, okay, vascular bundles are there, right? Surrounding this vascular bundles, you have what? Bundle sheath cells, okay. Surrounding vascular bundles, what do you have? Bundle sheath cells. So here if you see, bundle sheath cells also have chloroplast. Bundle sheath cells also have what? Chloroplast. This chloroplast is agranule. No grana is present. Agranule chloroplast. When there is no grana, there is no thylakoid membrane, nothing. There is no grana at all. That is, stalk of thylakoids are not present. It just has what? Stroma lamellae. Stroma lamellae is present.
Enzymes required for light independent.
Yes. So it has only stroma lamellae and stroma. Listen first. So here it is in mesophyll cells. In mesophyll cells, if you see, okay, it has what? Granule chloroplast. Granule chloroplast. So listen carefully beta. This is C3 plants. Surrounding the vascular bundles, there is only single layer of bundle sheath cell, yes or no? Whereas here if you see in C4 plants, 2 to 3 layers of bundle sheath cells will be present and there are no intercellular spaces in the bundle sheath cells. Okay? So there are no intercellular spaces. And their walls are impervious for gaseous exchange. Okay, their walls are with suberin depositions and it is impervious for what? Gaseous exchange. Means this bundle sheath cell will not allow any gaseous exchange between that. Remember this point. Okay, its cell wall is made up of suberin, which is impervious
For gaseous exchange.
Yes, for gaseous exchange. Fine. Now this concept is clear to you. Bundle sheath cells in C4 plants, these are the cells surrounding what? Vascular bundles. Vascular bundles in leaf are conjoint and closed. Okay. This vascular bundle is surrounded by what? Bundle sheath cells, 1 or 2 layers. This bundle sheath cells does not have any intercellular spaces. Wall is made up of suberin, which is impervious for gaseous exchange. Bundle sheath cells does not have grana. Okay, so it has agranal chloroplast and then stroma lamellae is present, stroma is present. This concept is clear to you, right? Now listen beta, this bundle sheath cells are connected with mesophyll cells by plasmodesmatal connections. Okay, bundle sheath cells are connected to mesophyll cells by what? Plasmodesmatal connections. Plasmodesmatal connections. So complete Kranz anatomy is clear to you, right beta? Any doubt anywhere? No, right? Yeah. So now if you see here, we will understand the mechanism of C4 cycle. Fine. Now it will be clear to you. Listen carefully. So here, zoom, okay. Okay, clear ma? Yeah. Listen. So first, in the gaseous exchange, carbon dioxide will enter into mesophyll cells array. This is clear to you, right? That carbon dioxide will be converted to hydrogen carbonate by carbonic anhydrase enzyme. Okay? Carbon dioxide which is entering into the mesophyll cells will be converted to hydrogen carbonate by what enzyme? Carbonic anhydrase. This hydrogen carbonate will be accepted by the primary acceptor, that is phosphoenol pyruvate. This phosphoenol pyruvate is 3-carbon compound. Phosphoenol pyruvate is what? 3-carbon compound. What is the primary acceptor in C3 cycle?
Ribulose 1,5-bisphosphate.
Good. What is the primary acceptor in C4 cycle? Phosphoenol pyruvate. Okay. And this is catalyzed by the enzyme pepcase, phosphoenol pyruvate carboxylase. So this has affinity only to bind with carbon dioxide, whereas there the enzyme which is catalyzing in C3 plants is rubisco, that has equal affinity to bind. So this is added advantage and adaptation of C4 plants. So this is catalyzed by pepcase and 1 carbon plus 3 carbon, forming out what stable product? 4 carbon oxaloacetic acid. This oxaloacetic acid is changed or isomerized to malic acid or ascorbic acid by the enzyme malate dehydrogenase. Okay. OA is converted to malic acid or ascorbic acid by the enzyme malate dehydrogenase. This malic acid or ascorbic acid will enter into the bundle sheath cells by plasmodesmatal connections. Can you see here the plasmodesmatal connections? Plasmodesmatal connections. Through this plasmodesmatal connections, what enters? C4 acid, that is malic acid or ascorbic acid. Aspartic acid. Thank you. So this is malic acid or aspartic acid which enters into the bundle sheath cells. Then this will undergo first, that is decarboxylation. Okay, 4 carbon compound will undergo decarboxylation forming out 3 carbon and 1 carbon dioxide. This 3 carbon pyruvate is regenerated back to the mesophyll cells. This regeneration is also through what? Plasmodesmatal connections. Okay. Now it is clear to you, of course, in bundle sheath cells which has agranule chloroplast, this is rich in what enzyme? Rubisco. This is rich in Rubisco. But then only gas which is entering inside the bundle sheath cell will be carbon dioxide, because no other gas can enter because its walls are impervious for gaseous exchange. Only 4 carbon compound will be entering into the bundle sheath cells, which is decarboxylated to 3 carbon and 1 carbon dioxide. This 1 carbon dioxide will conduct what? Kelvin cycle. Kelvin cycle or C3 cycle, fine, and rest 3 carbon will be regenerated back consuming 2 ATP. By what enzyme? Pyruvate phosphate dikinase enzyme. Okay. So clear with C4 cycle? Should I repeat it once quickly? Okay. Listen carefully. Okay. First, carbon dioxide, sit straight everyone. Carbon dioxide enters the mesophyll cell. Okay. It is first converted to what beta? Hydrogen carbonate. By what enzyme? Carbonic anhydrase. This hydrogen carbonate is first accepted by what? PEP, phosphoenol pyruvate, which is 3 carbon compound. Catalyzed by what enzyme? Pepcase. First stable product formed is, that is converted to as aspartic acid. By what enzyme? Malate dehydrogenase. And this is entering into the bundle sheath cells by what connections? Plasmodesmata connections. And decarboxylation happens inside bundle sheath cells. Decarboxylation happens, which forms 3 carbon pyruvate and 1 carbon dioxide. This 1 carbon dioxide will run what cycle? Kelvin cycle. Rest 3 carbons will be regenerated back to PEP into the mesophyll cells. By what connections? Plasmodesmatal connections. Fine, right? Is the enzyme. Fine. Now, in C4 cycle, for 1 carbon dioxide, okay, in C4 cycle, for 1 carbon dioxide, how much energy is being utilized? That is the question. Okay, energetics of C3 and C4 cycle. Listen first. Okay, for C4 cycle, for 1 carbon dioxide, fixation. Of course, carbon dioxide is fixed twice, but only 1 carbon dioxide is entering into the mesophyll cell. That we have to count. How much ATP is being utilized? 2 ATP during regeneration and 3 ATP during Kelvin cycle. Yes or no? So, in total, how much ATP beta? 5 ATP. How much NADPH? 2 NADH. Fine, clear, right? Next. So, for the formation of 1 glucose, the question comes like this. For the formation of 1 glucose, how many times carbon dioxide fixation has to happen? 6 times. So, it is 6 carbon dioxide and how much ATP beta? 5 into 6. How much? 30 ATP and then 2 into 6. How much? 12 NADPH. Sorry, NADH. It will not be in phosphorylated form. Fine, right? Okay. So, this is the energetics of C4 cycle. But you will get question not as C3 cycle and C4 cycle. You will not get like that. He will be asking you 2 different plants. Okay. How much extra ATP is consumed in sugarcane? How much extra ATP is consumed in sugarcane when compared to C3 plants during carbon dioxide fixation? If that is the question, what should be your answer? For 1 carbon dioxide, extra ATP consumed is how much? 2 ATP. For 1 glucose formation, extra ATP is? Glucose formation, extra ATP will be how much beta? 2 into 6. How much? 12 ATP is the extra ATP. quite a times in your NEET previous year questions about extra ATP the question is asked, and mostly you will get the examples of sugarcane, maize, sorghum. You can remember as SMS. Okay, sugarcane, maize and sorghum. Fine. Now C3 cycle, you listen. Energetics in C3 cycle.
Ma'am, in that tree also same ATPs are found.
Yes, same. Those are C4 plants, beta. More other plants are there, but these are most commonly asked questions. Most commonly asked examples in your previous year NEET questions. Okay. So in C3 cycle, for one carbon dioxide, how much ATP? 3 ATP and 2 NADH. Okay. For one glucose formation, yeah, 3 into 6, that is 18 ATP, 2 into 6, 12 NADH. Okay, and how much carbon dioxide fixation happens? How many cycles will happen? 6 CO2. This energetics is highly important. If you are having any doubt, just ask me now only. I will give you 1 minute time. Online students, you also can ask me better.
In which step those 2 extra ATPs are found?
That you have to say. You have looked into the previous cycle, no? You said you don't need any repetition also.
Pyruvate to phosphate are there.
That's it. You know the answer.
But only 1 ATP is.
No, Nana, it is 2. See, 2 ATP, okay? That is why have patience. Look into the things properly.
Ma'am, extra ATPs?
Hmm?
Extra ATPs?
No. See, extra ATP
Is
in comparison between C3.
and C4.
Clear, Sanya? So, any doubt anywhere in C3 cycle, C4 cycle and photorespiration? No, right? Now, CAM cycle is left, right? No issues. CAM cycle is same as that of C4. CAM cycle is same as that of C4, but I think I have no issues. Same as that of C4 cycle. The only difference is in C4 cycle 2 cells are involved. What are they? Mesophyll cells and bundle sheath cells. But in CAM plants, that is crassulacean acid metabolism, which occurs in succulent plants or desert plants, right? Like agave, cactus, opuntia, aloe vera. These are the examples, or some epiphytes as well. So this CAM cycle will occur only in one cell, that is mesophyll cell. Mesophyll cell. Okay, and here primary carboxylation or first carboxylation occurs during night time. Because of what? Scotoactive stomata. Okay, because of what, Nana? Scotoactive stomata. What is scotoactive and photoactive stomata? Any one of you?
Light flow
Mm. No. See, listen. So, scotoactive stomata is nothing but the stomata which opens during night time. Okay. Photoactive stomata is the stomata which opens during daytime. Okay. Scotoactive stomata opens during
Night.
Stomata opens during night time. This is an adaptation to prevent the process of transpiration in desert plants. Okay? This will prevent the process of what? Transpiration in desert plants. Clear, right? Any doubt anywhere here? No. Now you listen what happens. Okay. Stomata open during night time. Carbon dioxide enters into the mesophyll cells. Carbon dioxide will first convert to what? Hydrogen carbonate. By what enzyme? Carbonic anhydrase. Carbonic anhydrase. Okay? Yes, very good. Primary acceptor is what? PEP, PEP, phosphoenol pyruvate. Catalyzed by what enzyme? Phosphoenol pyruvate carboxylase. What is the primary product form? Oxaloacetic acid, which is changed to malic acid or aspartic acid. Malic acid or aspartic acid by what enzyme? Malate dehydrogenase. Okay, this will enter into the vacuole. There it is going to bundle sheath cells, but here it is entering into what? Vacuole, and this is stored inside the vacuole during night time. During daytime, this malic acid or aspartic acid will come out. This will be decarboxylated to 3-carbon pyruvate and 1 carbon dioxide. This 1 carbon dioxide will run what? Kelvin cycle. Fine. This 3-carbon pyruvate will be again turning to what? Regenerated back to PEP by consuming 2 ATP. And this Kelvin cycle, for 1 carbon dioxide it consumes 3 ATP plus 2 NADH. So the energetics of C4 cycle and CAM cycle are one and the same. What are the only differences, beta? Here it happens only in one cell, and first carboxylation during night time because of scotoactive stomata. Okay, any doubt anywhere in CAM cycle? If you have, you just let me know. Clear, right? CAM cycle, C3 cycle, C4 cycle and photorespiration. All of them are clear to you, right? With energetics. Okay. So, there is one homework to you which I want to give. In your NCERT there is a sheet, comparative sheet between C3 cycle, C4 cycle and CAM, that only beta. Yes. So, exercise that, and if you are having any doubts just let me ask tomorrow. Fine? Now, shall we go to factors affecting photosynthesis? Till here it is clear to you, no? Till here it is clear, right, ma? Now, factors affecting photosynthesis. Listen carefully. Factors affecting photosynthesis, this looks very simple, okay, but then questions from this area are mostly statement based only. Nowhere it is like a simple question. You look into all the NEET PYQs, question coming from factors affecting, though it is definitely statement based. So, you should have clear idea on the concept. Look at it carefully. This just takes 10 minutes. Listen carefully. Photosynthesis is the process by which the plants transform light energy to chemical energy. That is why we call it as what? Physico-chemical process. Physico-chemical process. Fine. And it is under the influence of several factors, be it external factors like sunlight, temperature, water, carbon dioxide, humidity, and internal factors as well, like amount of the chlorophyll, leaf size, leaf shape, this and all are internal factors. So, here comes a concept of Blackman's law of limiting factor. I will tell you what does this person said in the year 1905. Blackman's law of limiting factor says that any process, any biochemical reaction which is controlled by multiple factors, though the entire process is affected by that factor which is close to the minimal value. Understood? Not? Again I will tell, see, any biochemical process which is controlled by multiple factors, okay, the process is decided by that factor which is close to the minimal value. Okay, look at definition once.
Which is minimum, it decides the
Rate of reaction, or it decides that biochemical reaction. Whichever factor is minimal, that will decide the biochemical reaction. I will explain this with photosynthesis example. You will understand it more detail. Listen to it, ma. So, Blackman's law of limiting factor, if a chemical process is affected by more than one factor, then its rate will be determined by the factor that is nearest to its minimal value. It is the factor that is directly affects the process of its quantity is changed. That will directly affect the process and its quantity is changed. Once you recall the standard equation of photosynthesis,
the presence of sunlight.
okay, in the presence of sunlight and chlorophyll. Fine, right. So here what are the raw materials? Water, sunlight and carbon dioxide. So what are the external factors affecting the process? Carbon dioxide, water, sunlight, okay, and temperature also, because light intensity will decide the temperature. These are the external factors which are affecting the process. Amongst all this, let me put that question, let me see who will answer. Amongst all these factors, which factor is minimal? CO2. Is that accepted?
Yes.
Yes. Maximum atmospheric carbon dioxide concentration is how much beta? 0.03%. It can extend to 0.05, but extending maximum 0.05 though, it is damaging that chlorophyll organelle. It will damage the chlorophyll organelle. So which is minimal? Carbon dioxide is, this factor is minimal, and this factor will decide the rate of reaction or quality of reaction. After looking into the dark reactions completely, okay, C3, C4, CAM, C2 cycle, which is deciding the cycle, which is deciding the synthesis or fixation of glucose? Carbon dioxide. Okay, understood the example? Understood Blackman's law of limiting factor? One or two times in your NEET PYQs, the question has come from Blackman's law of limiting factor. Okay, clear with this na beta? No doubt nowhere? Yeah. So listen here, external factors. Okay, so external factors include light, carbon dioxide concentration, water, temperature and oxygen. Now, first let us look into light. Light can affect the photosynthesis process in three ways. Okay, first is light intensity, second will be light duration, and third will be light quantity. Yeah. Okay, light intensity, duration and quantity. So here if you see, the rate of photosynthesis increases the light intensity. If you have to look into this graph once, this graph where on x axis what is plotted? Light intensity. On y axis what is plotted? Rate of photosynthesis. So this light intensity which is plotted on the x axis, and on y axis what is given? Rate of photosynthesis. As the light intensity increases, rate of photosynthesis also increases. Yes or no? As the light intensity increases, what is increasing? Rate of photosynthesis increases. But then at the maximum increase, just at 10% of total sunlight, 10% of total sunlight, okay, this comes to a saturation point. Though you increase the light intensity, there will not be any increase in rate of photosynthesis, and that saturation point is attained just at 10% of total sunlight. Okay. So look at this theory, rate of photosynthesis increases the light intensity, increases as light intensity increases until it reaches to a saturation. This saturation occurs just at 10% of total sunlight. Blue and red wavelength are optimum for photosynthesis, right? Next, light duration does not affect the rate of photosynthesis, but it affects the entire process. Its effect is on whole, but it is not affecting the rate of photosynthesis directly. Amongst these three components, which component of light is directly having its effect on rate of photosynthesis? Light intensity. Simple, right? Understood? Understood this graph? Yeah. Next, carbon dioxide concentration. As I told you, carbon dioxide concentration is just how much? 0.03 to 0.04% in the atmosphere. Fine. If CO2 rate is increasing up to 0.05%, then it will increase the rate of photosynthesis. Increase more than 0.05, beyond this the levels can become damaging for longer periods. Understood? Next, so what is carbon dioxide concentration optimum for C3 and C4 plants? Before understanding this, let me recall you that point. In C3 plants, temperate, where carbon dioxide and oxygen are competitively are competitive with each other in binding with the enzyme Rubisco. Yes or no?
Yes.
So, this competition will increase the carbon dioxide concentration rate. Yes or no? More the carbon dioxide concentration, then only Rubisco will bind to carbon dioxide. So, here the optimum level of C3 plants carbon dioxide will be more. Fine. In C4 plants, if you see, as the enzyme is what? Pepcase. So, carbon dioxide binding with pepcase is unanimous. Fine. Carbon dioxide will ultimately bind with pepcase only. So, here carbon dioxide optimum will be less. Fine. In C4 plants, the optimum is around just C4 plants, it is 360 microliters per liter, okay, or 360 parts per million. Whereas in C3 plants, it is 450 microliters per liter or 450 parts per million. Okay. As the levels of carbon dioxide increases, the rate uptake also increases in C3 and C4 plants. So, these are what ma? Saturation points. Okay. The saturation point here, can you see, for C4 plants? Okay. C4 plants saturation point is what? 360 microliters per liter or 360 parts per million. And here can you see the saturation point? Yeah, 450 parts per million or microliter per liter. So, here the optimum for C3 plants is high because carbon dioxide and oxygen are competitively binding with Rubisco. That has raised the optimum range of carbon dioxide. Remember that. I am providing you reasons for each and every point so that you will remember the things easily. Okay. Now, but after a point, even as the levels of atmospheric carbon dioxide increases, the rate of assimilation or carbon dioxide fixation does not increase. Okay. Next. Water. Water is one more factor, external factor which is affecting the process of photosynthesis. So, water, you can consider it as water stress beta, water stress, which leads both to the turgid and flaccid conditions. Water stress. Any time there is any stress from external environment, what do? We will just shut the doors. The doors of a plant is what? Stomata. Stomata is the door of a plant. Whenever there is water stress, be it in turgid or flaccid condition, the immediate thing it will do is stomatal closure. Okay. This will prevent the exchange like carbon dioxide, and if it is in flaccid condition, drought condition, what happens? This results in wilted leaves, okay, which reduces the surface area and reduces metabolic activity. Water requirement in a cell will be reduced. Fine. This is the response of plant for what? Water stress. Understood? And then temperature. Okay. The light reactions are temperature sensitive, but they are affected to much lesser extent. Dark reactions are more temperature sensitive because, tell me two enzymes, Rubisco and pepcase. These two are the enzymes which are involved in dark reaction or light independent or biosynthetic phase. These two are temperature sensitive, remember that. So, the dark reactions being enzymatic, they are temperature controlled, okay, of which pepcase is cold sensitive enzyme. Fine. Pepcase means those are tropical plants, right beta, they are sensitive to what? Cold environments. Pepcase is what enzyme? Cold sensitive enzyme, remember that. Next, the C4 plants respond to higher temperatures and they show high rate of photosynthesis. C3 plants have much lower temperature optimum, whereas C3 plants temperature optimum is 20 to 25 degrees Celsius. C4 plants temperature optimum is 30 to 45 degrees Celsius. And then oxygen. Fine. So, there is some effect called as Warnberg effect, where you have to know about this. That is nothing but rise or increase in oxygen concentration when it is preventing any biochemical process, then it is called as Warnberg effect. This Warnberg effect you can see in which plants? C3 plants, because rise in carbon dioxide concentration will affect the fixation of carbon dioxide. Fine. So, here if you see, under conditions involving high concentrations of oxygen, Rubisco competitively binds with what? Oxygen, where and acts as what enzyme? oxygenase enzyme. This is known as what effect? Van Berg effect. This prevents rubisco from binding to what? Carbon dioxide. Hence, carbon dioxide fixation is significantly reduced. Eventually, rate of photosynthesis also is reduced. Understood Van Berg effect? Any doubt anywhere? No, right? Next. Now, we will look into the internal factors which are affecting the process of photosynthesis. Internal factors include number or arrangement of mesophyll cells, orientation of leaves, age of leaves, size of leaves, number of leaves, amount of leaves and photosynthetic products. Amongst
like sunken stomata.
yes, that comes here only in size, number of leaves and all. And here if you see, most important internal factors comes like amount of chlorophyll and photosynthetic products. Listen, number and arrangement of mesophyll cells. Mesophyll cell differentiation you will observe in dicot leaf. In dicot leaf, mesophyll differentiation is observed as palisade parenchyma and spongy parenchyma. Okay, palisade parenchyma and spongy parenchyma, which will increase the rate of photosynthesis. Whereas just spongy parenchyma arrangement is observed in what? Monocot leaf. Monocot leaf or isobilateral leaf. Only spongy parenchyma is observed. So, amongst these two leaves, where more carbon dioxide assimilation is seen? Dicot leaf. Remember this thing, carbon dioxide fixation, carbon dioxide assimilation or carbon reduction is one and the same. All the three terms is one and the same. Okay. What are they? Carbon reduction, carbon dioxide assimilation, carbon dioxide fixation, one and the same. Now, next is orientation of the leaves. Okay. Orientation of the leaves is better understood through phyllotaxy, that is arrangement of leaves. Few are arranged spirally like this, wherein all leaves will be exposed to sun. This will conduct more photosynthesis than normal arrangement of leaves. Okay. Next, age of leaves. When age of leaf is considered, then young leaves and old leaves, which leaf will conduct maximum photosynthesis? Mature leaf, because they are abundant in chlorophyll, chloroplast and mesophyll cells. Okay. Next, size of leaves. Which leaf? Bigger leaf will have more carbon dioxide fixation because it has more surface area than small leaves. Number of leaves. More the number of leaves, more photosynthesis. Next, amount of chlorophyll. Okay. So, have you observed any time about variegated leaves? Best example is croton. What is a variegated leaf? A leaf which has, yes, very good, a leaf which has green and non-green parts inside it is called as variegated leaf. Okay, which does not have much chlorophyll. When compared to green leaves, variegated leaves fixation will be low. Okay. So, that is best explained explaining the concept of amount of chlorophyll. Amount of chlorophyll is less in variegated leaves. So, there is less carbon dioxide fixation than in complete green leaves. Understood? Photosynthetic number is nothing but assimilation number, chlorophyll number or assimilation number or photosynthetic number is one and the same. This photosynthetic number is the amount of carbon dioxide in grams which is assimilated by 1 gram of chlorophyll in 1 hour. Okay. Just remember what is photosynthetic number, that is it. It is observed a direct relationship between the chlorophyll content of the leaf and rate of photosynthesis. Fine. More the amount of chlorophyll, more will be the rate of photosynthesis. Fine. And if all the other factors are favorable, increased chlorophyll will lead to what? Increase in photosynthesis. Fine. Last, photosynthetic products. Remember, any biosynthetic process will start to generate the product. Same the case of photosynthesis. All the raw material simpler products will combine to form the complex glucose molecule inside the cell. If the glucose molecule is already present in the chloroplast stroma, what is the necessary to start the reaction? Okay. So, photosynthetic products which are already formed will inhibit the reactants to start the reaction. This is called as feedback inhibition. Feedback inhibition. Same the case in respiration if we have to consider glycolysis. What is the end product of glycolysis? Pyruvate. If pyruvate is already abundant in the cell, it is already present, then what is the necessary to start the reaction of glycolysis, right? So, the products here are inhibiting the reactants, that is what feedback inhibition. Look at the theory ones. Accumulation of carbohydrates is called as what? Feedback inhibition of photosynthesis. Rate of photosynthesis will decrease because concentration of photosynthetic products in the cells increases the rate of respiration. Fine. More the amount of glucose, the energy from the glucose will be utilized by respiration. This has to be oxidized. Fine. So, this is all about the factors affecting photosynthesis. So, the complete gist of chapter, once sit straight in 2 minutes I will say what are the contents of this chapter beta. First is early discoveries, and then sequential reactions, both the photochemical phase, that is light reactions and dark reactions. Light reactions, 3 steps. What are they? Absorption of light in the form of photons, then photolysis of water, formation of assimilatory power. There we have learned the concepts of cyclic photophosphorylation, non-cyclic photophosphorylation, chemiosmotic hypothesis, and then after that biosynthetic phase we have started, that is light independent reactions, where we have learned what? C3 cycle, C4 cycle, CAM cycle. Only cycle which is operated in C3 plants is C2 cycle, that is photorespiration. After the biosynthetic reactions, what did we learn? Factors affecting photosynthesis. We have learnt about external factors and internal factors. External factors are what? Light, temperature, carbon dioxide and water, oxygen. And here we have learnt the concept of Blackman's law of limiting factor. For oxygen we have learnt Van Berg's effect, and amongst the internal factors, age of the leaf, shape of the leaf, orientation of the leaf, and most importantly amount of chlorophyll, photosynthetic number, assimilation number, lastly what? Feedback inhibition by photosynthetic products. This is the complete chapter beta. From this chapter what type of questions you can expect? Any type of question like statement based, assertion reason, match the following type of questions. Very good. Most importantly numerical questions from the energetics of biosynthetic reaction. From the energetics, most importantly what you may get? Numerical questions, and from chemiosmotic hypothesis as well. Fine. So, exercise all the bioenergetics of this chapter as well. Fine. Any doubt anywhere, to let me know. Online students also just drop a message in chat box ma, any doubt anywhere, I will be clarifying that. So, that is all for today's session, and thank you.
Is it